Hello, everyone. I am Yojana Gadiya, a PhD student at University of Bonn and a data scientist at Fraunhofer ITMP in Hamburg. Today, I will talk about pharmaceutical patent landscaping, which is an approach for understanding how patents can be leveraged for drug discovery. We start by talking about what a patent is. So, a patent is a formal document that discloses the invention to a public. This formal document provides legal protection for the innovation and the discovery that is done. Furthermore, it grants market exclusivity rights for about 20 to 25 years to the patent owner. There are a number of offices where you can file these patents. In the United States, you can file it in the USPTO. In the European continent, you can file it in the EPO, while in the Asian continent, you can file it in the JPO or other regional patent offices. As you can imagine, these are formal documentations, so they are mostly written in free text and hence are non-machine friendly documents. So when uploaded on databases and made publicly available, they are usually PDF documents. The patent document is used for a number of scenarios currently. The first one being portfolio management, where you can look at the patent portfolio of a given company and see how many patents they file per year. You can also do something called as patent landscaping or trend annotation, wherein you look at the number of patents for the company has filed for a particular compound and see how many of these compounds have actually reached the market. From the chemistry and the drug discovery science side, you can look at patents for chemical space exploration. That means that if you have a potential lead molecule, you would want to find out how the patents around this lead molecule look like, and also find out strategies which are called as freedom to operate, the FTO strategy, to identify how you can optimize the compound for your use case, such that the compound itself is novel and is outside the scope of the existing patents. To extract patents for drug discovery, we created a tool which is called as the Patent Enrichment Tool, short acronym called PEMT. This is a Python-based tool. And what the tool does is extract patents for a given indication area. So this could be target agnostic, where you start with the target and look for active compounds based on bioactivity data available in public database such as Campbell. Or it could be from the compound side, where you already have a pre-lead and want to find out patents that are relevant for drug discovery. In either of the cases, once you have the compound, what we do is look into Shaw-Campbell, which is a public patent database, and extract and filter out patent documents that A, are active, so they are between the 20 to 25 year period, and are talking about drug discovery. That means that they talk about the formulation of a drug or the utility of that particular compound based on a target or an indication area. Once you collect all of these targets, there are a number of things that you can achieve. First, you can identify drug targets that are currently being pursued by industry and academia partners and find potential collaborators, for example. B, it's a one-stop shop for collecting patents relevant to indicating to an indication or a target area, rather than from a chemical-centric approach, which is the classic way of finding patents. So here in the heat map, you can see that we have aggregated patents for Alzheimer's disease based on the different targets and have a target portfolio for each of these. So for different companies. So in a way, this is the target portfolio for an indication area for different pharma players. Last but not the least, this patent aggregation technique provides a real-time assessment for finding and identifying non-interesting targets or compounds that are there in the pipeline. In the figure on the right-hand side, you can see there is the historic patent landscape of one of the targets called as Adam-17. And around the year of 2005, you see that there's a downfall in the number of patents that were there for this target. And this downfall is directly corresponding to the failure in the clinical trial that took place around the same year. As a result, there is a close correlation between the patents and the activity that happens in the patenting world with respect to the clinical trial information. In summary, what the PEM tool allows is to extract patents for a given indication or a target area of interest and do a patent landscaping approach. In this patent landscaping approach, you can identify multipurposing of existing patents. So reusing a particular patent for a compound in a different indication area, for example. You can identify cost reduction strategies where you look into which patents should be prioritized and which should be abandoned based on the market space. Lastly, you can formulate a patent portfolio aggregating all of the patents over the years for a given company. There are a number of future perspectives to this application. First, that we can include and add biologics, which are currently excluded in consideration. Next, you can, the whole pipeline relies heavily on the bioassay and bioactivity data that is in Chemble. This can be expanded to bioassay data available in different public resources. Lastly, from the tool, what you get is a time-dependent and active patents only, and it lacks giving you the whole perspective of legacy patent documents, which are currently non-active or have been abandoned at some point. And this would be one of the future perspectives we can develop the tool wherein we include all of the non-active patent documents as well. With this, I would like to end my presentation. Thank you for listening.